Alright guys, here we go. The first part of chapter 7 was about types of cells and the organelles for a eukaryotic cell. So we did that in class, so I'm hoping you guys review your notes because you're going to have a quiz tomorrow over the first part of the chapter and 7.4 and 7.5. So here we start off with 7.4. You'll have an additional lecture 7.5, which is separate from this one. And also throughout the lectures from this point forward, I'll be reviewing stuff that you'll need for EOC, or I feel that may be important for you in the final, okay? So 7.4, Cell Systems 1, Nuclear Transport. So as we know already, the nucleus is the control center of the eukaryotic cell. The inside part of the nucleus is very, it's highly organized. Here you have DNA and DNA production, because DNA has to be replicated in order for cells to come from pre-existing cells. You also need RNA. RNA is used for protein synthesis. You have RNA production, and then you have the nucleolus, which is responsible for ribosome assembly. And we'll get more into detail throughout the lecture. Here's the nucleus in itself. The nucleus is a membrane-bound organelle. That means it has a membrane around it. Here's a nucleolus. The nuclear envelope is made up of two membranes and a nuclear pore. Alright, so this is your eukaryotic cell. Here you have the mitochondria, which is also a membrane-bound organelle, the Golgi apparatus, the ER, and if it has ribosomes on it, it's the rough ER. If it doesn't, it's a smooth ER. Okay? Then lysosomes, peroxisomes, and everything else you learned in, or we went over in class. When we discussed organelles. Alright, here's the nuclear envelope. It separates the nucleus from the rest of the cell. And the components inside the nucleus from the rest of the cell is composed of two membranes. So here's the first membrane, and here's the other membrane. Here you have the inside part of the nucleus. Here you have the cytoplasm, but in the book they call it the cytosol. And here you have the nuclear pore complex. Alright, the nuclear pore complex. Okay? And it's a double membrane barrier. It extends, the nuclear pore complex extends through the two membranes, as you can see here, and they're actually latched on or attached to the nuclear pore complex. Connects the inner nucleus also with the cytosol, and it's made up of 30 different proteins. So remember, proteins are made up of amino acids. So there's 30 different proteins or polypeptides that make up this complex. Polypeptides or proteins are made in the ribosomes. Ribosomes are the protein factories. Just a little review there. What travels out of the nuclear pore complex? So DNA cannot travel out of the nuclear pore complex. It stays within the nucleus, first of all, because of its size, and then many other reasons. So what it does, it synthesizes or it makes RNA. So there's three types of RNA. You have mRNA, tRNA, and rRNA. mRNA is a messenger RNA, is the actual message or the copy, a single-stranded copy of the genetic code. Alright, tRNA is used to make transfer RNA, which carries amino acids to the ribosomes during protein synthesis, and then rRNA, which is also known as ribosomal RNA, makes up the ribosomes, which are assembled in the nucleolus. So what can travel into the nuclear pore complex? You have nucleotides, both DNA and RNA. Remember, deoxyribonucleic acid and ribonucleic acid, two different sugars. Remember, the nucleotides have also a phosphate sugar in the base, and the nitrogenous base is different in both. DNA has GCAT, guanine, cytosine, adenine, and thymine, and RNA has guanine, cytosine, adenine, and uracil. Alright, and also what travels into the nuclear pore complex are the proteins needed for the synthesis of RNA and DNA. Here is another model of the nucleus, and just to give you an idea of what the nucleus does and how it works, the membrane works, or the nuclear envelope works. The nucleus imports about 500 molecules through each pore. All right, there's about 2,000 to 5,000 pores in the nucleus, and this occurs every second. So 500 molecules through each pore every second, and there's about 2,000 to 5,000 pores. All right, how do molecules move into the nucleus, move into the nucleus. All right, small molecules like nucleotides move in freely. All right, just remember that the nuclear envelope is also selectively permeable. Only those molecules tagged or selected to move into the nucleus are allowed to move in, or are also allowed to move out. But this also requires some energy. All right, it requires some energy to move molecules in and out. How do molecules move into the nucleus? These molecules that have to be tagged before they moved in. It's like a zip code, as they mentioned in your book. So the zip code is a tag, and it's made in the ribosome. All right, it's a polypeptide, a chain of polypeptide, a chain of amino acids. The nuclear pore complex reads it and allows them to come in. And as you can read in the book, one of the key things you have to read is a nuclear localization signal. All right, you have to make sure you know that for your quiz tomorrow. So now, what I want you guys to do is summarize your notes for this lecture. And when you're summarizing your notes, I want you to answer this question: how are the nuclear membrane and cell membrane similar, or different, or different? All right, let's make that and or different. Or different. So this is it. It was a short lecture. Make sure this lecture is not a substitute to reading the book. So make sure you read section 7.4, 7.5, and go over your organelles for your quiz tomorrow.